All right, welcome, folks. I think it's about time to get started, right? It should be 2:30 more or less, so I guess I'll just go first. Just let everyone settle down. All right, cool. Start my timer. All right. Hello, folks. My name is Martijn Kissmer, and today I'm talking about the role of structural variation during adaptation to a stressful host and a seed beetle. This way, cool. So evolutionary rescue is the case in which populations that otherwise would go extinct persist because of genetic variation. This is becoming an increasingly documented process, and one of the things that we're primarily noticing is how fast of a process this can be, with a lot of cases of this happening in 20 generations or less. This is particularly interesting both because of the applied sense, right, figuring out things of whether or not species can survive climate change, finding out whether or not bacteria will develop antibiotic resistance, but it's also fascinating from a theoretical perspective because it first of all challenges the paradigm that evolution is a slow-acting process, and it also allows us to study evolution within our lifetime, or within a PhD. And so, experimental evolution, wrong slide, sorry, I was reading this. So my big questions for today are, what are the genomic mechanisms of evolutionary rescue, and how repeatable are the mechanisms of evolutionary rescue? With the bigger picture insights of gaining insights into the mechanisms that promote adaptive evolution. And so for this study, I leveraged experimental evolution, which is a really great approach for asking these kind of questions, primarily because the drivers of selection and the demographic histories of your populations are both known. And additionally, the environment can be tightly controlled to create similar conditions across replicate populations, which is really great for studying parallel evolution. My study system is cowpea seed beetle, Calucibrochus maculatus. This is an agricultural pest. It's a seed beetle, and so what they do is they lay their eggs on the bean. The larvae burrow in, they eat the contents, they emerge as adults. Now, interestingly, cowpea seed beetles have been found to colonize multiple hosts in agriculture, most notably green lentil. Green lentil is interesting because initial survival on it is 1 to 3%. It's incredibly low. It's not a nutritious resource. Yet, populations reared in the lab on green lentil have been able to completely recover their survival and fecundity in less than 20 generations. So this is an extreme case of evolutionary rescue, and it's one that we've documented quite well in the lab. However, the genetic mechanisms have not been fully explored. So our collection consists of 4 separate populations from Burkina Faso, South India, Brazil, and California. And these different backgrounds can be used to determine whether or not repeatability is robust to different sources of standing genetic variation. All right, so before I got here, previous folks in the lab did an adaptation experiment where they took several replicates of our Brazil, California, and Burkina Faso populations, and they adapted them to lentil over the course of 20 generations. This was an evolve and resequence experiment, so they took DNA both before and after, and they did pooled whole genome sequencing. I came in about a year ago, and I realigned the data to a novel reference genome that had chromosomal level resolution, and I calculated 10 kilobase FST windows. All right, so just to break down what's going on here, we have 6 panels of FST. The columns represent replicates from the same geographic origin, and we have 3 different geographic origins. The red bars here designate regions that are shared for being highly differentiated from the same geographic origins. And the first thing that you'll notice is that every single population has regions that are shared among chromosomes 1 and 9. So in these regions, natural selection seemed to act in a very parallel manner. It shaped differentiation very similarly in those regions. You'll also notice that there are a lot of other peaks that are not shared, right? There's a lot of regions that have high FST, other spikes, and the reason we think we see those is because there was such a strong bottleneck at the initial aspect of adaptation here, right? Survival of 1 to 3%, and so there's a lot of things that may have risen in frequency stochastically. And so to try to get around that, I created back-cross introgression lines. And to create these, I took our lentil lineages and our cowpea lineages, I hybridized them, I put the offspring on lentil. Once they emerged, I again back-crossed them back to the cowpea lineage and repeated this until we see what we see on the right over here. And so the end result was a population whose genome most closely represents the cowpea lineage, except for regions that are required for survival on lentil. I did this, and I did GBS sequencing on these populations, again made 10 kilobase per FST windows, and here's our first example. So as you can see, a lot of the peaks on the bottom here, and just to explain this, these are 2 FST plots. This plot is FST between the original parental lines, the cowpea and lentil. This plot is FST between the cowpea and the introgression lines. And as you can see, all the spikes in regions that didn't experience adaptive differentiation, so everything outside of these red shaded regions, lost their peaks. Everything on chromosomes 1 and 9 that had adaptive genetic information, supposedly, kept that differentiation in the back-cross introgression lines. Now to show you how this looks in the rest of our populations, again, you can see it's highly parallel. All of our Burkina Faso lineages maintained the differentiation on chromosomes 1 and 9, whereas every population maintained the differentiation on chromosome 1. And this is super cool, right? This is 10 independent cases of adaptation where the same supposedly causal variant in different genetic backgrounds ended up on the same chromosome. So in this case, adaptation seemed to be highly parallel. Now this was interesting, and I wanted to look more closely into what was going on. And so I focused my efforts on chromosome 1. And the first thing that I noticed, again, is this huge block of FST. It's a 110 megabase per chromosome. The entire thing seems to light up in some of our populations. And so the first thing I thought was maybe structural variation, right? Is there some large structural variant that might be driving adaptation? And to get at this, I used an Oxford nanopore. I generated long-read sequences, which are particularly good at finding structural variants. I did this on our introgression lines as well as their parental lines. And as you can see here, I identified a 20 megabase pair structural variant right over the region of high differentiation. This is FST zoomed in on chromosome 1. It goes from, again, 64 to 84 megabase pairs right over that region. This is extremely interesting in the context of rapid evolutionary rescue because structural variants can facilitate rapid adaptation, right? They could do this primarily through two ways. They can lock adaptive variants onto a single inherited block through reduced recombination. And additionally, they can introduce adaptive variation at the breakpoints, right? If you think of, for example, a part of the chromosome switching, they can essentially remove genes, duplicate genes, whatever else. So this is extremely exciting in the context of witnessing adaptive evolution. This potentially might be what we're seeing driving the adaptation forward at such a pace. And so to get into this a bit further, I also revived a previous study where folks in the lab used our mung adapted lines. And mung here is as suitable as a host to the beetles as cowpea. So they adapted these mung adapted lines over to lentil over the course of this many generations. You see the plot from the beginning here. You can see that they recovered their survival rate within 15 generations. They've been maintained ever since. And the folks from this study created F2 back crosses. So what they did is they hybridized the mung lineage with the lentil lineage, and then they back crossed that F1 back to the mung line. They then measured development time to adult and adult mass in these F2s and sequenced them with GBS sequencing. I took this data, realigned it to the novel reference genome, and used a Bayesian sparse linear mixed model through Gemma for trait mapping of development time and adult mass. Now, crazy enough, here's the trait mapping. Here's the posterior inclusion probabilities along with the PVE. Minus that one point for lineage 1 and chromosome 1, every single point that potentially could be explaining genetic variation for development time and adult mass falls on that region where the structural variant is located in our other experiment. And even more interesting, it's the exact same SNPs that show up for both, which means that there's a single peak falling under our structural variant that seems to be driving both development time as well as adult mass. And if you look at the PVE, for the most part, it's fairly high. We have 50 to 70% with a couple exceptions. But I do truly believe that there's a decent bit of genetic variation for these traits, and it seems to map right to those regions. So looking a bit more closely, I decided to examine exactly where these peaks were falling. And the main peak at 83 megabases falls on a transcriptional regulator ovo-like isoform, which is the protein we see on the right here. This is a zinc finger transcription factor that's involved in development. That, again, is extremely compelling for rapid evolutionary rescue, right? Thinking about the fact that these transcription factors, gene regulatory genes, might be influencing downstream phenotypes that might explain why they're able to adapt so quickly. The next peak at 81 megabases is a glucose dehydrogenase, which catalyzes the oxidation of glucose and is involved with metabolic processes. And then finally, we have a sub-peak at 79 megabases, which is a mucin 4-like isoform. And mucin 4 is involved with the development of mucosal linings in the intestinal tract, and it essentially protects it from debris and from pathogens. So now I'm not totally sure yet whether or not these three are some way causal or if they're all just in linkage with something that's right in that region, because this is all within 4 megabases. At the same time, it would make sense given the fact that we're looking at a nutritional-based form of adaptation that occurs during the developmental stages for a beetle. On top of that, they are also all linked to the same structural variant. So there's a potential chance, like we talked about earlier, that they might be all locked in a single inherited phenotype. I also took some time to look at the RNA expression analysis that was generated for this study. Again, align that to the genome, the differential expression analyses. I'm not going to talk too much about this one, but I do want to focus on gene 1408 right here, the one that has the highest negative log 10 p-value. If we plot the location of that locus, it falls right on the FST spike in chromosome 9 for our other study. Even more interestingly, I looked at the function of this protein, and it is a signal transduction protein that's involved with cell proliferation, cell growth, cell survival. And so it's another gene regulatory protein, which is super cool. And it almost makes me wonder or not the transcription factor we found in the previous study in any way is influencing the transcription amount here. But that's food for thought later. All right, so main takeaways from here are that evolutionary rescue shaped genetic differentiation in a parallel manner. Adaptive regions were consistent among replicates. A large structural variant underlies the adaptive region on chromosome 1. This region mostly seems to explain the genetic variation in development time and adult mass. And finally, regulatory genes may play a large role in evolutionary rescue, which is something that I would like to investigate in the future. All right, you will notice that I do have more time, and that is because I also want to talk about a method here that I'm in the process of developing. It's not totally there, but I want to bring it to you guys' attention, get the idea out there, and also see if people want to come up with some ideas in the future. And so, I've been working on an idea to try to find the needles in the haystack here, right? We're looking for causal variance in regions of high LD or potentially on structural variance. This is difficult because you have a giant wall of FST. It's kind of hard to find outliers. For example, chromosome 1 is a great place to study this. It's a 110 megabase per long chromosome with about 1,100 genes, and approximately 200 of those genes are found on our structural variant of interest. So I've been coming up with the idea to try to use AlphaFold, which is an algorithm developed by Google's DeepMind to potentially predict the 3D structure of proteins based on their amino acid sequences. Now, the idea here is to essentially fold every protein on the chromosome for our populations and try to find out whether or not there's a structural difference in the proteins, both in the pre-adapted lines and in the post-adapted lines, to try to find out whether or not there's actual structural differences based on the DNA sequences. And just to give you a quick overview of how that works, I took our pooled sequence data from the beginning of this experiment. I created alternate reference genomes that insert their variants. I then used a genome annotation to extract the exons and coding regions, and use an open reading frame finder to find out the regions that are likely translated into proteins. From there, we have our translated amino acid sequences, and I did this, again, for each gene, for each population. So I have 10 populations, 10 variants of each gene. I folded every single one of them using AlphaFold. And from there, I had all of our protein predictions. I used a TM score. A TM score is the standard score for basically comparing the 3D structure of proteins to search for protein structure diversity both within host treatments, so within the copy lineages, and then between host treatments, so between the copy and lethal lineages. And this is the part where I'm still working on it, so if any discussion later on in the conference would be greatly appreciated. But I plotted the 95th percentile of outliers for that over on the chromosome here. And just visually speaking, it looks like they might be clustering in locally high areas of FST, but I wanted to be extra sure, so I ran a bootstrap over here. And what we see here, these red lines correspond to the 95th percentile of windows of FST that deviate from the moving average, right? So these are regions that are not necessarily high for FST across the chromosome, but high in FST for their local region. I then plotted the location of these genes, which are the black lines, and counted within the closest 10 windows of FST how many of them are red, how many of them fall in these locally high peaks of FST. Sum that up for each gene, and then sum that up for every gene in the set to arrive at the number we see here. And then to compare that against the distribution, I ran a 10,000 replicate bootstrap where I did the same thing with 10,000 replicates of the same number of genes selected from the genome. And as you can see here, we have a distribution. And it's not significant. It's not in the 95th percentile, but it is marginally different from the mean at the 90th percentile. So I do think we are picking up some signature. What I think we're picking up now is more so the inherent, there's genetic diversity there. It's translating into difference in amino acid diversity that isn't related to adaptation in our case. And that's why we're seeing hits that seem to correlate with genetic differentiation. But I think that genuinely, if we do sort through the cycle more, there might be some promise. And just to give some evidence for that, I ended up finding the protein structures for our transcriptional regulator that we found earlier in this talk for both the copy lineage and the lentil lineage. I aligned them visually. You can see the alpha helix here that's pointing right at you. That's where the zinc binds for the zinc finger. And not only do the structures look visually different, but I aligned the TM score, and they have a score of 0.4, which, considering the fact that they're the same protein, is incredibly low. So I do think there's a signal to be found here. I would love to talk to people about ways to sort through it better. All right. For acknowledgements, I'd like to thank all the folks in the lab that made this possible, all the people who conducted the previous studies, and my funding sources. If you have questions or want to hear this explained more slowly, send me an email. I think we have time for one or two questions.
When you calculated the TM score, did you only use the structured regions, or did you also include all the unfolded spaghetti there?
So I tried it both ways. I computed a confidence metric to specifically isolate those regions, and then I also worked with the entire protein, and the entire protein seemed to have more consistent results. Yeah, yeah. Thanks. Oh, one more.
One observation is that AlphaFold in its current incarnation is not great at predicting structural consequences of point mutations,
right.
But it will probably improve so it will be worth revisiting.
Totally, and that's part of the reason why I wanted to get this out there. I think it's a good one to think about. Great. Any other questions? Well, thank you so much, everyone. Appreciate it.
Welcome, everybody. My name is Martijn Schenkel. I'm a postdoc at the University of Groningen. And today I'll give an overview of some of the postdoc work I've done with Manus Patton at Georgetown University and Arvid Ogren, who's at Cleveland Clinic Foundation, on the paradox of the organism. Doesn't work. Okay. So I want to start off actually with the observation that when we look across the tree of life, we see individuals in all sorts of shapes and forms, ranging from unicellular prokaryotes to multicellular eukaryotes to complex eusocial groups that consist of multiple individuals. And when we think about this, this kind of makes sense because all of these kind of individuals represent biological hierarchies. So they're nested hierarchies of elements that cluster together into higher order collectives. For example, genes cluster into chromosomes, which cluster into genomes, etc., all the way up to biological groups of individuals. And that is usually explained in the concept of major transitions, where lower-lying particles or lower-lying evolutionary agents come together to form collectives. And those collectives come to be evolutionary individuals in their own right. So they come to respond to selection and develop adaptations. And what I want to work to today is what we can actually do to revert these major transitions. So is there some process by which these evolutionary products or these evolutionary individuals can be torn apart into lower-lying evolutionary individuals as they once were? So basically to establish what is a major reversion. And the rationale for this is that when we look across, or if we look at the process of adaptation, which is the theme of this session, I suppose, is that all of these collectives that can respond to selection to some extent and develop adaptations have to grapple with internal conflicts. And so for organisms, these internal conflicts can give rise to all sorts of stuff that interfere with their biology, such as self-genetic elements in the form of post-segregational killers or imprinted genes that affect the allocation of resources, and also on a cellular level in the forms of cancers and microchimeric cell lineages. So there's a bunch of things inside organisms that can interfere with their function and their capacity to be adapted and suitable for their environment. And so we published a paper last year where we kind of analyzed how these different suborganismal elements may interfere with organismal biology, identifying two main strategies, which we dubbed transmission distortion, which, for example, for genes is basically interfering with the rules of Mendelian inheritance, or trait distortion, where some selfish elements act to basically alter the organism in some way as a whole to kind of get its way on in the grand scheme of evolution. And we can put all sorts of selfish elements in this kind of diaphragm, like, okay, some of these are more trait distortion-y, some of these are more transmission distortion-y, some of them are both. And we can also kind of think about how badly they affect the organism as a whole. So we can think about their impact on organismal fitness or the capacity of the organism to function as an evolutionary agent, and kind of make some guesswork, some verbal arguments, and say these are not so bad and these are completely terrible. Something that we've been recently focusing on, and I'll make sure you don't focus on the math, is to kind of come up with a way to think about quantifying these arguments in a more rigorous way. And some recent work that we're now refining in response to some very helpful criticisms in peer review. So we developed two metrics, which we dubbed trade unity and fitness unity, that the first of which, trade unity, actually focuses on the alignment of these different parts, these different cells or genes in an organism, and asks how aligned are those optima that these genes or cells may have relative to each other. And that basically deals with kind of the conflict over the phenotype that may arise within an organism. The second one is fitness unity, and that basically revolves around the evolutionary impact of those internal conflicts. So how deleterious are these different conflicts to the organism as an evolutionary agent, or otherwise, how fitness optimal is an organism at the various optima of those different parts? And so coming to the point of my talk, so the idea of a major reversion, we've been asking ourselves, when do we actually undo these major transitions? So when are these internal conflicts so disruptive that an organism doesn't really qualify as an adaptive evolutionary agent anymore? So when should we actually talk about them as sort of collections of competing parts that use the organism as a temporary vessel to get their way? We can kind of rephrase that question by saying, okay, can these internal conflicts give rise to selfish elements and lead to organismal biology being so disrupted that they're actually torn apart, resulting in a major reversion? And if so, what does that even yield? So that was the question we had at first, like what does that even mean? And the simple way to think about that is to think about, for example, a multicellular organism falling apart and giving rise to single-celled descendants. So a multicellular ancestor gives rise to single-cellular descendants. But then we started thinking about, okay, major transitions, major reversions, what is the point of these changes in evolutionary individuality? And we came up with a bunch of criteria that we want to assign to a major reversion. So in order to talk of a major reversion proper, it needs to meet certain criteria. And we see some of those criteria across different selfish elements and different forms of life. So paternal sex ratio chromosomes, for example, destroy the entire genome that they are transmitted with. So they leave the core genome of an individual is not transmitted, only that chromosome is transmitted. So basically, if you have PSR, this paternal sex ratio chromosome, you have no evolutionary success. Other examples include rogue worker bees that reproduce on their own. We see transmissible cancers, which are cancerous lineages that are transmitted between individuals. HeLa cells, which are basically immortal single cells in a sense, but they just don't live outside of the lab. And possibly very interesting viruses, which are basically free-living genetic elements. And so coming back to that previous framework that we developed for organismal unity, we can think about how they might actually help us think about when major reversions could occur. So if we are, in Evolution 101, we think about organisms as all things serve the being, so genes and cells always work for the organism. We have high fitness unity and high trait unity. Nothing is really going wrong. Then we can have low trait unity. So okay, we can agree some internal conflicts exist within organisms, and yes, they might not agree over phenotypes or transmission or whatever, but it doesn't really matter. So yes, you can have some selfish genes, but in the long run, they don't really negatively affect the organism in any meaningful way. Then we get to the quadrant that's probably, for technical reasons, impossible, and I'd be glad to discuss that in detail later. And finally, when we have low trait unity and low fitness unity, we basically get to the realm where we have a lot of disagreement over organismal phenotypes, and these differences in optima are also meaningful. So if you are residing at the optima of a specific selfish element, for example, that leaves you very poorly off as an organism. And it's maybe in this situation that we can get major reversions. But that's still a kind of hypothetical scenario, just saying, okay, there could be some evolutionary pressure, but do we actually have a pathway for these major reversions to occur? Think about that, we get to the following schematic. So we started off with a situation where we have no internal conflict. So we see this organism, which is the pink circle, contains 3 pink dots, and those pink dots are all the same. Then we get some internal conflicts where these different dots come to follow different selection, or come to be affected by different selective pressures. So they get different evolutionary optima that they might want to pursue. And so we can see, for example, in this case, that the pink dot is still at its fitness optimum, but the blue and the yellow one are not. We can then get to a case where we get some adaptive evolution of one part in this collective that basically distorts the biology to such an extent that some of these parts are now really poorly off. So really, they're very far away from the fitness optimum. And depending on how they can actually evolve, it might actually run into a case where it's easier to evolve to just abandon this collective and go and do something on your own. So this is the actual part where we get the major reversion happening. So this is where particles leave their collective and become individuals once again. We can get the actual establishment of that major reversion. So now we've kind of separated off this blue particle from its former co-particles in the pink collective, which is now yellow. And we might get some later evolutionary responses in that residual individual that brings it back to something less bad for the pink particle in this case. Now, of course, the question then becomes like, okay, if we assume this to be valid, why haven't we actually seen any major reversions? Why don't multicellular organisms give rise to single-cell individuals all the time? And that's a very valid question, and there's a bunch of valid answers to that, I think. And that basically, like, okay, reversions might be physically inaccessible. So we can think about, for example, a mitochondria might not be able to abandon its host cell. It might not be able to live freely on its own anymore. Or maybe it can, but it's just so rubbish at performing in the wild outside of its host cell that it's just not viable or not competitive. So they might leave, but they don't become evolutionary individuals in their own right. For other reasons, they might just be difficult to identify. So maybe we don't really accurately assess what has happened in the evolutionary history. And of course, a lot of people have kind of worked on the assumption that these major transitions are irreversible. So have we even been looking for them in the first place? Or is this something that we've just been not thinking about at all? So wrapping this up, we could basically conclude over these 3 papers that we've been working on that selfish elements disrupt organismal function through transmission and or trait distortion. Organisms are not unified agents of evolution, but they have internal conflicts that they must grapple with in order to achieve their goals. And when we think about the evolution of individuality, major reversions are one unexplored concept, but there are a lot of more other questions out there that we have to grapple with when we think about how individuality evolves. And so I want to end off with some acknowledgments to collaborators, particularly Manis and Arvid, as well as funding from the John Templeton Foundation, the ADF, and ESIP. If you like talking about this stuff, come to Groningen on October 14 and 15, where we have a workshop which is sponsored for one of the special topic networks. And I'll end with shameless advertisement for one of my collaborators, Naomi Greenberg, who will give a talk at 4:15. And we have a symposium on Tuesday, the whole day in 520D. And I'll happily take questions.
All right, hello everyone. My name is Javad. I just finished my master's with Sally Otto's lab, with Sally Otto, and I'm going to tell you about what I did during my master's. But first I'm going to tell you a little bit of a background of how it started. So in our lab, Penny, another PhD student who is also giving a talk on Tuesday evening, is doing this evolution experiment where she adapts these to lethal concentrations of different heavy metals, and then she tests their fitness in different environments, for example, in the absence of metal. And one thing that she consistently sees is that metal-adapted lines grow slower in the absence of metals compared to their ancestor. And there are multiple reasons for that. One of them is that different metabolic pathways have different susceptibilities to different metals, and what is optimal in the presence of metal is not necessarily optimal in the absence of metal. And another reason for that is that some of them have, sometimes these have some adaptations to, for example, pump out heavy metals. And those adaptations require allocation of resources. And allocation of resources can have some cost for reproduction because cells have limited resources. And when they allocate resources to those adaptations, they have less resources to grow. And so yeah, that's what I define as cost of an adaptation, the reduction in birth rate of an organism due to allocation of resources to an adaptation. So I became interested in studying that. And so so to give you a more concrete idea of what is a resource, let's focus on this example. Let's say we have some cells that are dealing with a toxin, and they can have 2 hypothetical adaptations that help them deal with the toxin. One of them is making chelators, which are some proteins that bind and neutralize the toxin, and they prevent the harm. So making chelators requires making proteins, which needs amino acids, energy, and action of ribosomes. On the other hand, let's say cells can do some pumps that they already have more active to pump out the metals, and that only requires allocation of energy. So then I asked myself, okay, these are different resources. How can we compare cost of one type of resource to the cost of another type of resource? So that was my first question. And then I also asked myself that, okay, if the reason that there is a cost is that resources are limited, what happens if we give cells more food? Does that increase the cost of those adaptations or decrease the cost of those adaptations? And so I started reading, and so there are 2 of the main frameworks that have been developed to investigate cost of adaptations are energy allocation framework and ribosome allocation framework. A good example of energy allocation framework is this paper called The Bioenergetic Cost of a Gene by Lynch and Marinoff. And so they say that cost of a gene depends on the fraction of cells' energy that is allocated to production of a gene and its product. And so when they do that, they are basically implicitly assuming that cells are energy limited, and they have a limited energy budget, and they are allocating it between different adaptations. And they also have to translate cost of all adaptations into energy equivalence, which is, for example, they have to calculate okay, this amino acid is 20 ATPs. The other framework is the ribosome allocation framework, which has been developed by bacterial physiologists. So that one of the main things that they have been observing for a long time is that cells that are, when we give cells better food and they grow faster, they also have a higher RNA to protein ratio, which is a proxy for how many ribosomes they have. So, and because they have been observing this linear increase in the fraction of ribosomes in the cell as we give cells better food, they concluded that ribosomes must be very central in cell reproduction because, most of the cell is proteins and ribosomes are making those proteins. And so when they want to consider cost of adaptations, they ask, okay, what fraction of cells' ribosomes are allocated to make proteins that are required for this particular adaptation. And so to compare these two frameworks, let's focus on this schematic cell. So when we have a cell, food and nutrients need to first be uptaken, then catabolism breaks down food into energy and like some metabolites, and those metabolites are later, they become converted to building blocks of macromolecules, like amino acids and nucleotides. And then energy, some of it is used for maintenance requirements, and some of it is used to convert those building blocks into macromolecules. And macromolecules are then degraded with some rate. Some of the substrates, some of those building blocks are recycled, and some of them are lost. And so energy allocation framework focuses on that like energy production pathway, and is kind of assuming that that's what's limiting cell reproduction. On the other hand, the ribosome allocation framework is mainly focusing on protein production. And so it doesn't explicitly consider energy limitations that cells face. And so I wanted to combine these two frameworks and come up with a framework that allows us to simultaneously consider limitations at the level of energy allocation and ribosome allocation. And so to do that, first I simplified things a bit. Well, like I group all of the reactions in like uptake, catabolism, and anabolism into a single reaction, which I call provisioning, and that converts food and nutrients into substrates, which are basically energy and amino acids and other building blocks. And so some of substrates are used for maintenance requirement, and the rest of them are converted into macromolecules by this second reaction called biosynthesis. Then macromolecules are degraded as before, and like there is some of the substrates are recycled and some of them are lost. So I divide macromolecules of the cell into three fractions. Provisioning fraction are the macromolecules that are facilitating the first step of provisioning. Biosynthesis fraction are the macromolecules that are facilitating the second step. And then the rest of macromolecules are structural fraction, which don't facilitate any of those two steps. So I just defined provisioning efficiency as how fast are the provisioning fraction macromolecules, and like biosynthesis efficiency as how fast biosynthesis fraction macromolecules are. And so the next step is I need to find a way of calculating burst rate. So for a cell to be able to replicate, it needs to double its biomass. So I'm going to assume that burst rate of a cell depends on the rate at which it can double its biomass, which is basically rate of biomass production minus biomass degradation divided by biomass of the cell. And so that equals this thing on the right, which I explained. So it equals phi b, which is what fraction of macromolecules are doing biosynthesis, times epsilon b, which is how fast they are working when they are saturated, times gamma b, which is how saturated they are. And how saturated biosynthesis enzymes are depends on how much biosynthesis enzyme we have and how much provisioning enzyme we have, minus degradation rate. So there is a trade-off between the size of biosynthesis fraction and its speed. So here, the purple line is the size of biosynthesis fraction, and the blue line is the speed of biosynthesis fraction, and burst rate is just the purple line times blue line. In this graph, degradation is ignored, and it's assumed to be 0. So as we can see, when biosynthesis fraction is very small, although biosynthesis enzymes are working very fast, there is very few of them, so burst rate will be low. When there is a lot of biosynthesis enzymes, phi b is large, but then they will work very slowly, so burst rate will be low again. And there is an optimal size of biosynthesis fraction which maximizes burst rate. And so if cell is under optimal allocation or like somewhere close to that, like we observe like this distribution of macromolecules between three different fractions. So as I said, a structural fraction is assumed to be fixed, and then cell adjusts the allocation between provisioning and biosynthesis to maximize burst rate. And so the only thing that I need to do to be able to calculate the optimum is to come up with a function that describes how saturation level of biosynthesis fraction changes as its size increases. And so I approximated using this function. I don't get into the details of why it's exactly a good function, but I would be happy to discuss that. But basically what this function is saying is that when the rate of substrate supplied by provisioning, maintenance demand, and like recycling is way larger than what biosynthesis fraction can consume, biosynthesis enzymes will be saturated, and when it's the opposite, the saturation level approaches 0. And using this, I can investigate the effect of changing different parameters on the shape of these curves, which is basically our fitness surface and the optimal allocation. For example, in the left, you can see that when I decrease the biosynthesis efficiency, the optimal size of the size of biosynthesis fraction under optimal allocation increases to make up for the reduced efficiency. And so I can calculate burst rate analytically when we are under the optimal allocation. And that equals to this equation, which I'm going to walk you through. So burst rate under optimal allocation equals to investment into growth, which is 1 minus a structural fraction, or basically what fraction of macromolecules are allocated into provisioning and biosynthesis, times return on investment into growth, which is the average efficiency of provisioning and biosynthesis fractions, but not average exactly. It's like it has some sort of average. So that's our baseline. Then we have some loss due to maintenance demands, and that loss is maximal when A is large, and A is the extent to which provisioning, which is the first step, is limiting. So if provisioning efficiency is way smaller than biosynthesis efficiency, A approaches 1, and then loss due to maintenance is maximal. And when provisioning efficiency is way larger than biosynthesis efficiency, so the second step is limiting, then the extent to which provisioning is limiting approaches 0, and allocating to maintenance isn't super costly. And the loss due to degradation is the opposite. If provisioning is limiting, it's not that costly. If biosynthesis is limiting, it's very costly. And so using this, I can try to answer the original questions that I have, which was how can I compare the cost of making chelators and higher activity of pumps, and how those costs will change if I give cells more food. So chelators are some proteins that don't participate in either provisioning or biosynthesis pathways. So they are kind of a structural. So making more chelators is like increasing the structural fraction. And the cost of increasing a structural fraction depends on RI, or return on investment, which is the average of provisioning and biosynthesis efficiency. So if I give cells more food, RI is going to be larger, and making chelators is going to be more costly. And the reason for that is that when environment is good, it's very costly for the cell to not allocate its macromolecule to growth and allocate those macromolecules into something else. Because if those macromolecules were allocated to, let's say, pumps and ribosomes, like they're allocated to making ribosomes and other metabolic enzymes instead of chelators, they could have made a lot of biomass. But if environment is really poor, that cost is lower. On the other hand, higher activity of pumps requires more energy, which is like increasing maintenance demands. And the cost of that depends on the extent to which provisioning is limiting, which was the first step. And when we increase the food quality, that first step, provisioning, becomes less limiting. Therefore, cost of allocating energy to these adaptations decreases. So, I mean, this was what I was trying to answer at the beginning. How can I compare the cost of higher activity of pumps and like making chelators, and also how those costs will change when we change the environment. But I'd like to end my talk with two slides that are kind of adjacent. So first of all, I assume the allocation is optimal, but that assumption is not necessary. My model is basically describing the fitness surface and predicts how it changes under different conditions. And also, another thing that I want to talk about is that I talked about cells, but I'm curious how useful this model can be in helping us understand the growth of other organisms. For example, think about an ant colony. It also wants to increase its biomass, and also it's allocating its individuals between provisioning, which is the individuals that bring in food, structural, like the individuals that are soldiers, they're kind of structural, and like queen is maybe doing more like biosynthesis, making new individuals. Or like in an organism, it's allocating its tissues between provisioning, which is like allocating it to like guts and stuff. And like some things are structural and some things are biosynthesis. But I mean, I'm curious, I'm not saying that this is a good framework to think about that, but I think it might be helpful if the key assumptions are met. And those key assumptions are that, first of all, fitness depends on growth rate, and growth rate is determined by biomass accumulation rate. These are the main assumptions that I made in my model. And also I assumed that like that saturation function has a specific form. So yeah, with that, I'd like to thank Sally and everyone else in my lab, and thank you for your attention, and I'll take questions.